saw a moment ago, this kinesin molecule is capable of walking along this microtubule. So the idea here is that it's kind of like a wire. So the analog would be, back here, in this example, this is somewhat analogous to wireless communication. So a molecule can be released here, propagates in free space, and hits the receiver. So this is somewhat wireless. Whereas when I have a microtubule connecting the receiver over here, and a transmitter over here, and a receiver over here, and some message-bearing kinesin molecules propagating down that microtubule, this is somewhat analogous to wired communication. So in other words, instead of electrons propagating down the wire, now we have kinesin. Again, Sanjay did an excellent job describing how kinesin walks, but just briefly to remind you, this represents a microtubule, and basically what will happen is one side of the kinesin will remain anchored, whereas the other one swings around and basically walks like that. One thing that's important in the context of this work is that depending on how all of the parameters work out, these walks are polarized. They can either occur in this direction or in the other direction. So this particular method, there are actually two ways in which this active transport can occur. This we call rails down. So in other words, this microtubule is acting like a rail, and this kinesin molecule is acting like a locomotive traveling down that rail. One thing you can do with this locomotive is that you can actually attach a cargo to it, and that's something that we're going to look at in future work, is that you can actually attach a message-bearing bead to this kinesin and have it propagate down the rail. One thing you can do with this, you can actually send like a packet like you would do in the internet, except it's an actual packet that you're attaching to the kinesin. But this is the alternative method, and this is the one that we're going to examine in this work, which is called engines down. So instead of the kinesin propagating along the microtubule, in fact now we have a bed of kinesin down here attached to some kind of substrate, and the microtubule is actually propagating along that bed of kinesin. So whereas the other case was analogous to a railroad, this is more analogous to a conveyor belt. So basically what we do is we drop the microtubule on the bed of kinesin, and the microtubule moves. So instead of the kinesin carrying our message, now it's the microtubule. And again, depending on how this microtubule is dropped on the bed of kinesin, it can either propagate this way, or it can propagate the other way, and that's not something that can be especially well controlled. So the main idea here is that we're looking at microfluidic lab-on-chip devices which are lined with kinesin. So these microchannels will be lined with kinesin, and the idea is we're going to drop microtubules into those channels and allow them to propagate from one end to the other. So what we're going to do is we're going to look at how these devices, we're going to simulate how these microtubules propagate, and secondly, we're going to investigate how to send messages with these microtubules and what the ultimate limits on those messages are. So we can simulate how these work. So this is what I'm about to show you is a simulation of microtubule motion in a cigar tube shaped microtubule. So what you're going to see is 3 microtubules simulated to be released at intervals of 1 second, and they will propagate from one end to the other of the device. So there's the first one, which is blue, second one, which is red, and third one, which is green. Remember, they were emitted at intervals of 1 second. Let's try that again. So you see the red one, the blue and green microtubules initially started out propagating in the direction of the destination, whereas the red one started propagating towards the back wall of the microtubule, or excuse me, of the channel. Another thing that's interesting to note here is that although they're emitted in the order of blue, red, green, they arrive they arrive in the order of blue, green, red. So they actually arrive out of order. So that actually causes huge problems for analysis of the communication system. So in order to understand how this acts as a communication system, let's just briefly review the abstract information theoretic model of communications. Basically, we have a message, we transmit the message, there's some distortion that occurs in this channel, and then the receiver's job is to figure out whether, is to try to make its best guess of what the message was, and hopefully that the messages agree on both sides. If they don't agree, then we've made an error. So the idea is we want to minimize the probability that an error occurs. So now we have to decide what constitutes the signal in this channel and what constitutes the distortion. We are going to examine a very simple system in which we send, basically we send a binary signal, or a binary vector. The signal consists of a 1 corresponding to a release of a microtubule, or a 0 corresponding to no release. So in other words, if I want to send you a 1, I drop a microtubule and that propagates to you. And if I want to send you a 0, then I don't do anything, and hopefully you will not observe anything. And this will occur at intervals. So every t seconds, I will make a decision to either release a microtubule, which is a 1, or not do anything, which is a 0. And then your job is to guess what I sent. So we've got two forms of distortion in this channel. The first one is the random arrival times of microtubules. So I drop a microtubule into my channel, it propagates to you, it arrives some random time later. So for example, if my interval between bits is very short, it's possible that, as we saw in the example, as we saw in the simulation, it's possible those bits will arrive out of order. So in other words, you'll have a tough time figuring out what order these bits arrived in, and you might actually make errors trying to decode my message. The second form of distortion here is the fact that all the microtubules are indistinguishable. So although I colored them in the simulation for ease of telling them apart, if nothing's attached to the microtubules, you generally can't tell you can't tell them apart. So once they all arrive, you have no idea what order they were sent in. So that's basically what you're observing. If you think of this from the perspective of a traditional communication system, you're not observing the channel outputs. What you're observing is the channel outputs in the order they arrive. So it's like you're sorting them and then trying to unsort them to figure out what was sent. So the first source of distortion, the random arrival time. Here what I'm showing you is simulated arrival time distributions. So this basically tells you the PDF, the probability density function of particle arrivals for various lengths of the channel. So this is for 10 micrometers, 20 micrometers, 30 micrometers. For 10 micrometers, this is the PDF that we get. It's bimodal. The bimodal feature of this arrival PDF arises basically because you can either start propagating directly towards the receiver or in the opposite direction. So if you start propagating directly towards the receiver, you get this PDF. And if you start propagating directly towards the back wall, basically it has to turn around, and you generally get arrivals that are a little bit later. As the channel gets longer, these become a little more diffuse. And furthermore, the latency is increased, obviously, because the propagation time